Bismillahirrahmanirrahim. Assalamu alaikum. Dear students, this is Zokia Tariq, and today we are going to study the enzymes. The word enzyme is derived from Greek word enzymes. Enzymes mean leavened or ingest. Leaven basically means, or leavening is basically means the rising of bread, that is basically caused by yeast. What are enzymes? As we have studied earlier, that enzymes are made up of proteins. They are basically 3D structure of protein that can be tertiary or quaternary structure. What do you mean by tertiary structure and quaternary structure? Basically, tertiary structure of a protein means that if a protein structure is made up of one polypeptide, and quaternary structure if the protein is made up of two or more than two polypeptide chains. So enzymes are globular proteins, like you can see in the diagram. This is an enzyme which is made up of protein units, and all these units you can see on the screen are basically the polypeptides made up of amino acids, and this space here is known as active site where a substrate can attach. Basically, enzymes act as catalyst, and these can accelerate the rate of reaction. We know that in our body there are multiple reactions are taking place at a moment. These are called biochemical processes or biochemical reactions. These biochemical reactions are involved in metabolism of the body, and metabolism is basically categorized into two categories, mainly into anabolism and catabolism. Anabolism is the assembly of smaller molecules into larger macromolecules, and catabolism is a breakdown of those high molecular weight bearing macromolecules into smaller units. So during these reactions, a high amount of energy is used to start these reactions. So enzymes act as catalyst to speed up these reactions, and by lowering the activation energy, the energy used to activate a chemical process. During the biochemical process, enzyme is used, but it remains intact, and it can be used again and again when the reaction is proceeded at or ends. If we talk about the structure of enzyme, basically enzyme consists of an active site that may act as a catalytic site or binding site. A specific substrate of that enzyme may bind to the active site and is converted into the product. So enzymes are specific for what they will catalyze. Like lactase enzyme only will catalyze the lactose. Enzymes are reusable because, as I already told you, enzymes are inert. They cannot change or destroy during the reaction. Most of the enzymes end with ASE, like sucrase, lactase and maltase have end word ASE. Basically, ASE is derived from diastasis, which means separation. How do enzymes work? What is the mechanism which enzymes use to convert the reactants into products in a biochemical reaction? Enzymes work by weakening bonds which lower the activation energy. So how this activation energy is lower? Because enzyme weaken the bond. For example, in the case of sucrose, when a enzyme have to break the bonds between sucrose molecule it will weaken the bond between the sucrose and fructose molecule. This is the activation energy lowered by the enzymes. So, why reaction can easily converted into the products. This is the example without enzymes and with enzymes. Why we need enzymes and what is the significance of enzyme? This is the best graph which shows how enzymes are important for reaction. When a reaction is in progress and reactions are converted into products, if we don't use an enzyme, then there will be activation energy up to this high, and if we use the enzyme, this activation energy drop from here to here. So the reactants can easily convert into the products as activation energy is lowered from this place to this place. As we can see in the diagram, there are two test tubes here. The test tube A, there is no enzyme, only the substrate H2O2. This is the substrate. And in a test tube B, substrate is with enzyme. So the products are formed H2N plus O2. So the bubbles are formed, you can see in the diagram. And what was the substrate? What was the enzyme here? Enzyme was the liver homogenate that contained catalase enzyme. Basically, catalase enzyme converts the substrate, this particular substrate, into the products. Enzyme substrate complex. In the mechanism, when a substrate and enzymes join to form the enzyme substrate complex, the substance or the reactant which is binded with the enzyme is called substrate and form the EH complex. Basically, the active site is the restricted region of enzymes where the substrate bind. So it acts as binding site as well as catalytic site. When the substrate are bounded to the active site, it is then catalyzed into the product. There are two models which present the relationship between substrate and enzymes. These two models are lock and key model and induced fit model. Lock and key model was proposed by Emil Fischer in 1890 to visualize the substrate and enzyme interaction. According to the lock and key model, active site is not flexible. So when a substrate is attached to the enzyme, no changes occur in the substrate and it remains intact. But according to the induced fit model, which later was proposed by a scientist Koshland in 1959, which was modified form of that lock and key model. According to the induced fit model, a change in the shape of enzymes active site occur because active site is flexible part of enzyme. So when a substrate attached to that particular active site, so conformational change occurs or induced by the substrate on the active site. As you can see, the substrate have a smaller difference than the active site. So when the substrate attaches to the active site or binding site, so the active site changes its shapes according to the substrate. There are many factors that affect the enzyme's activity. This can be environment, this can be cofactor or coenzyme, or enzymes inhibitors. For efficient activity, enzymes need cofactor and coenzymes, and enzymes are also affected by environment, for example, pH, temperature, or substrate enzyme concentration. Enzymes are also affected by inhibitors, environment factor. temperature, pH, and substrate concentration. If we look at temperature, we know that temperature provides the kinetic energy. Heat provides the activation energy, and chemical reactions are accelerated at high temperature. When temperature is lower than the optimum temperature, reaction cannot proceed. The temperature on which an enzyme can maximally work or work at their best is called the optimum temperature. For example, most of the enzymes in the human body work best at 37 degrees centigrade. When the temperature is increased from 37 to above, as enzyme is made up of protein, so the temperature will denature the enzymes. So denaturation after 37 and less speed and less fast reaction before 37. pH is also another important factor which affects the rate of reaction. If we lower the pH of environment, it may also affect. For example, most of the human body enzymes work at 7 pH for their maximum activity. So we can say that for those reactions who work best at 7 pH is the optimum pH for those reactions. As with the substrate, it is also another important factor that affects the enzyme activity. If we increase the substrate concentration after the saturation point, no change in the activity will happen as there is no active site remain empty. So the rate will be constant after a certain limit. At this point, almost all the active sites are occupied and enzyme is saturated here. The other factors are cofactors and coenzymes. Basically, cofactors and coenzymes used for the efficient working of enzymes. Basically, cofactor is a non-protein part of enzyme which is essential to work enzyme. Cofactor can be detachable, called activator, and it can be covalent bond with the enzymes called prosthetic group, or may loosely attach with the enzymes called coenzyme. Inorganic substances, for example zinc, iron, and vitamins, are sometimes needed for proper enzymatic activity. For example, hemoglobin needs iron to pick up the oxygen in the blood. The last factor which affects the enzyme is called enzyme inhibitor. Basically, enzymes can counter with two types of inhibitors, reversible or irreversible. As the name indicated, the reversible inhibitor can be detached from the enzymes by increasing the substrate concentration. This means that there is a weak bond between the reversible inhibitor and enzymes. But if the inhibitor is covalently bonded with the enzymes, it cannot be detached from the enzyme and completely block the active site. Reversible inhibitors can be categorized into two more types: competitive inhibitor and non-competitive inhibitor. Basically, competitive inhibitors are chemicals that resemble an enzyme's normal substrate and can compete with it for the active site. So the competitive inhibitors attached at the active site, while the non-competitive inhibitors do not enter the active site but bind to another part of enzyme, causing the enzymes to change its shape, which alters the active site. Basically, competitive inhibitors is attached at another site instead of this active site. So when it is attached to the other site, the shape of active site is changed. So a genuine substrate could not attach to the enzymes. So this is how the enzyme inhibitor affects the rate of reaction or enzyme activity. Take home assignment for you. Find the enzymes from internet which are involved in our daily routines, for example in digestive system. Write down their names and functions. What is the optimum temperature and pH for the body? And I have mentioned this over the lecture. Thank you for listening. If you have any question, please ask in the comments.